All right, let's jump right into this deep dive. Today, folks, we're tackling something pretty essential for you as medical microbiologists.
Oh, absolutely.
The biosynthesis of polytide antibiotics.
Okay.
We're getting all our information straight from Chapter 16 of Antibiotics, Challenges, Mechanisms, Opportunities
Great source.
by Walsh and Wenswich. You know, have you ever felt like those tiny factories that just churn out antibiotics like they're operating in some language you don't quite get?
Right?
Well, we're going to break it all down for you today, how these polytide assembly lines actually function.
Yeah.
And honestly, the similarities to processes happening in your own body might surprise you.
Well, it's true. The way that these microscopic factories create these polyketides shares some really close similarities to how our own cells make fatty acids. But there's a catch, right? There's always a catch. There are some really interesting little twists and turns that make these pathways capable of producing this huge array of powerful antibiotics.
So it's like you're recognizing familiar machinery, but then someone's given them a whole new set of blueprints.
Yeah.
So walk us through how these building blocks come together.
So think of it this way. Both polyketides and fatty acids are basically put together from this very simple starter unit, acetate.
Okay.
And they both use this neat trick of forming what we call beta-ketone intermediates, which are kind of like the foundation of their structure, molecular structure. And just like with non-ribosomal peptide, NRP, antibiotics, which we've talked about before, they use this modular organization, so it's like an assembly line, right, really carefully put together, where specific enzymes are like little workers, each one doing their job to link these building blocks together.
So we're talking a highly structured process here, not just molecules randomly bumping into each other.
Precisely. But there's one key difference that really sets polyketides apart from those NRPs.
Okay.
And it's this. During chain elongation, polyketide syntheses, they create carbon bonds, whereas NRPs, they rely on nitrogen-carbon bonds to link their building blocks together.
That seems like a subtle distinction, but I imagine it has huge implications for the kinds of structures they can create.
Yeah, it does.
So how did these intricate polyketide assembly lines even come about?
Well, that's where this really interesting evolutionary connection comes in. Polyketide syntheses, or PKSs, they actually evolved from fatty acid syntheses. So these are the enzymes that are responsible for making those essential fatty acids that we all know and sometimes love to hate.
The building blocks of those energy-storing molecules that are constantly making headlines in the nutrition world.
Exactly. So let's kind of dive a little deeper into how these cellular factories actually operate. In fatty acid synthesis, there's this molecule called malonyl-CoA, and it steps onto the scene.
Okay.
And it provides these precious carbon atoms that are needed to actually extend that growing fatty acid chain.
So malonyl-CoA is like the fuel,
Yes.
the raw material that keeps the assembly line going.
That's a great analogy. And the engine that drives this whole process forward is a chemical reaction called malonyl-CoA decarboxylation.
Okay.
Now, it sounds complicated, but basically, malonyl-CoA loses a carbon dioxide molecule. And this releases energy, and it helps form that crucial carbon bond through what we call a thioclase condensation. Now, to keep everything running smoothly, those growing fatty acid chains are tethered to these acyl carrier proteins, ACPs. Now, these act like those little magnetic paper clips that keep papers organized on a busy desk, right? It prevents these intermediates from diffusing away and just makes sure it's a really efficient process.
So they're like the organizers, the ACPs of this whole operation.
They are.
And I remember you mentioned that fatty acid synthesis involved this whole reduction process, right?
You are correct. Each fatty acid chain undergoes what we call a 4-electron reduction cycle. So think of it as it's gaining electrons to become fully saturated, meaning it has all the hydrogen atoms it can possibly hold. And this process, it needs the help of two very important molecules,
Okay.
NADPH and FADH2, which act as electron donors.
Got it. So it's like this carefully orchestrated dance of molecules with malonyl-CoA bringing those building blocks, the ACPs keeping everything in check,
Right.
and NADPH and FADH2 providing those essential electrons.
Perfect. Now, remember how I mentioned that polyketide synthesis has a couple of these little twists and turns compared to fatty acid synthesis?
Yeah.
Well, that's where things got really exciting.
Okay, I'm ready. Tell me about the twists.
Okay. A key difference is what we call incomplete reduction. So in fatty acid synthesis, that beta ketone group that forms in each step is fully reduced, like we just talked about.
Right.
But in polyketide synthesis, this reduction can be incomplete.
So some of those beta ketones might hang around instead of always being transformed.
Exactly. This incomplete reduction means that the final product, that polyketide molecule, can have these beta ketone groups, or they might be modified in other really creative ways.
Yep.
And that, my friend, is one of the secrets to this incredible diversity we see in polyketide structures.
So it's like having more options on the menu.
Yeah.
You can stick with the classic, fully reduced product, or you can spice things up, keep some of those reactive groups around.
That's a great way to put it. And to make things even more interesting, PKSs, they have this trio of enzymes that they can use to really control the level of reduction and basically fine-tune the final structure of those polyketides. These enzymes are called ketoreductase, KR, dehydratase DH, and noyl reductase ER.
So they're like specialized tools in the PKS toolbox.
You got it. And whether these enzymes are present or absent in specific modules within that PKS assembly line, it determines what that final outcome will be. So for example, if the KR is missing, that ketone group, it stays there. If the DH is absent, that beta hydroxyl group, that OH, remains. And if the ER isn't doing its job, you get a double bond in that polyketide chain. So it's like this really carefully choreographed dance where each enzyme has a very specific role to play in shaping the final structure.
It's fascinating. It's like having a set of switches that you can turn on or off to create all these variations.
Exactly. And those variations, those tiny little structural differences, are what make polyketides so incredibly potent and so diverse in their antibiotic activity. It's what allows them to target different bacterial processes and really overcome those resistance mechanisms.
That's incredible. Now, you mentioned something earlier about polyketones being unstable.
Yes.
So what happens to them if they're just left to their own devices?
Those mischievous polyketones. They have this tendency to cyclize, meaning they form rings. And this brings us to another really key aspect of polyketide diversity, the formation of macrocycles.
Macrocycles. Are those related to the macrolide antibiotics I've encountered?
You are sharp. Macrolactones are these large wing structures, and macrolide antibiotics are indeed a really important example. So erythromycin, a very classic macrolide, it boasts a 14-membered macrolactone ring.
So we're talking some serious molecular architecture here.
Absolutely. Now, before we dive into the specifics of how those rings are formed, we need to talk about how these PKS assembly lines are organized, because there are actually two main types, type I and type 2.
So different management styles in these molecular factories.
Great way to put it. So type 2 PKSs, they're like a network of independent workshops.
Okay.
So each catalytic domain, those little workstations that we talked about earlier, they're located on a separate protein. So it's more of a decentralized approach. You have these different enzymes coming together to assemble the final product.
Interesting. A more collaborative approach. What about type I?
So type I PKSs, they're more like those big, massive, centralized factories where all the domains are all fused together on a single protein. Think of it as an all-in-one solution.
So type 2 is all about teamwork and flexibility, while type I is about that streamlined efficiency.
Exactly.
I bet these organizational differences have a big impact on how those macrocycles are assembled, right?
Absolutely. But before we get there, there's one more layer of classification that we need to consider within those type I PKSs.
Okay.
They can be iterative or modular. So iterative type I PKSs, they use the same set of domains multiple times, like a skilled worker who can handle different tasks on the assembly line. This is actually commonly seen in fungal PKSs.
So a jack-of-all-trades approach.
Precisely. Now, in contrast, modular type I PKSs, they have a dedicated module for each step in that whole assembly process. Each module, it's used only once as that polyketide chain is passed down the line. Like, imagine a really specialized factory, like those for making complex electronics.
Got it. So iterative is about multitasking, while modular is about specialization.
Exactly. And a perfect example of this modular system is the biosynthesis of erythromycin.
Okay.
That very macrolide we discussed earlier, it's a classic example of a modular type IPKS in action. And that is actually where we'll pick up in part 2 of this deep dive.
Awesome. All right, folks, we are just getting started. Make sure you tune in next time as we break down these assembly lines step by step and uncover the secrets behind those potent macrolide antibiotics.
It's gonna be good. Welcome back to our exploration of polyketide antibiotics. Remember we left off talking about those fascinating macrolide antibiotics and how their assembly lines are organized?
Yes. Those macrolides with their impressive ring structures. You were about to tell us how those rings come together using erythromycin as the example, right?
Exactly. So erythromycin, with its 14-membered microlactone ring, is produced by a modular type I, PKS, often called DEBS, that's short for 6-deoxyrethronyl B synthase. And this incredible molecular machine, it's got 7 modules, each one dedicated to a specific step in the whole assembly process. And they're actually spread across 3 proteins.
So it's a true division of labor.
Yeah.
With specialized modules all working together in sync.
Precisely. And here's the really cool part. The presence or absence of those tailoring enzymes, the KR, DH, and ER within each module, actually determines the final structure of that erythromycin molecule.
So it's like a code embedded within the PKS that determines which chemical groups are added or modified at each step.
Brilliant analogy. For example, module 4 of DEBS, it lacks all 3 tailoring enzymes. So that beta-ketone group from that step, it actually remains in the final erythromycin structure. But in module 7, the last one in the assembly line, all 3 enzymes are present, so you get complete reduction at that position.
So each module has its own personality based on which enzymes are active.
I like that. Now, once the erythromycin molecule is all assembled, it still needs to break free from that PKS, right, and form that characteristic ring structure.
Right.
And this is where a special enzyme comes in, a macrocyclizing thioesterase, or TE. Think of it like the conductor that brings the whole orchestra together for the grand finale.
So the TE is responsible for both releasing the molecule and shaping it into that final form.
Precisely. It's like a skilled sculptor chiseling away to reveal the masterpiece within. So the TE, it catalyzes the formation of that macrolactone ring by first excluding water from the active site. Then, it very carefully positions a specific hydroxyl group on that growing polyketide chain so it can attack the thioester bond, and this forms the ring and sets the molecule free.
It's amazing how such a tiny enzyme can orchestrate such a precise and crucial step.
It really is. It's a true testament to the elegance and efficiency of these biosynthetic pathways. Now let's contrast this very elaborate modular assembly line with the production of another well-known antibiotic, tetracycline.
Okay, it changes scenery. What makes tetracycline synthesis so different?
Well, remember those type 2 PKSs we talked about? The ones organized like those independent workshops?
Yeah.
Well, tetracycline biosynthesis relies on this simpler system. It uses a single ACP, a single KS, and a chain length factor, or CLF.
So a smaller team working on this one.
And here's the key difference. Those tailoring enzymes, the KR, DH, and ER, that played such a big role in erythromycin's assembly, they're often missing in these type 2 PKSs.
So it's like skipping the quality control checks along the assembly line.
In a way, yes. And this lack of tailoring leads to the formation of these polybeta-ketone chains. And if you remember, those are quite unstable and prone to cyclization.
Right, those mischievous polyketones always wanting to form rings.
Exactly. And that's precisely what happens in tetracycline biosynthesis. These reactive polyketones undergo a series of spontaneous cyclization reactions, and that's what leads to that characteristic four-ringed structure of tetracycline.
So even though the assembly process is simpler, the final molecule is still pretty complex.
That's the beauty of these systems. They achieve complexity through all sorts of different strategies. Now, to illustrate how this all comes together, let's zoom in on the biosynthesis of a specific type of tetracycline. Oxytetracycline.
Okay, sounds good. I'm ready to get into the details.
Excellent. So the oxytetracycline assembly line kicks off with a malonyl-CoA starter unit, a modified version of our friend malonyl-CoA.
Okay.
And through a series of 8 condensation reactions, it builds up this long chain with 9 ketone groups.
9 ketones. That sounds like a recipe for chaos.
You're right. It's a very reactive intermediate, just primed for those cyclization events. But here's where it gets really interesting. An enzyme called oxygenase steps in, and it specifically reduces one of those ketone groups, the one at position 9, to an alcohol group.
So oxygenase is like a strategic intervention guiding that cyclization in a specific direction.
Precisely. This reduction is crucial because it sets the stage for the next key step. An enzyme called oxyK, a cyclase, comes in, and it catalyzes the formation of the first ring of the tetracycline molecule, the D ring.
So we're starting to see those ring structures emerge.
Exactly. And remember, this is a type 2 EKS, so it's a collaborative effort. Other enzymes, like OxyN, they join the party and catalyze the formation of the B and C rings through a series of reactions.
It's like a team of skilled artisans, each one contributing their expertise to create that final product.
Perfect analogy. And finally, that fourth ring is formed, the molecule is released from the ACP, and voila! We have pretremed, the first soluble intermediate in the whole pathway.
From a simple starter unit to a complex tetracyclic structure, that's quite a journey.
And it's not over yet. Remember those post-assembly line modifications we talked about?
Yeah.
Well, they play a crucial role in transforming this inactive pretetramid into the potent antibiotic we know as oxytetracycline.
Right. It's like adding those finishing touches that bring the whole artwork to life.
Exactly. In oxytetracycline, we see a series of methylations, hydroxylations, a transamination, and a final reduction, all meticulously orchestrated by specific enzymes.
So it's not just about putting the core structure together. It's about refining it, adding those crucial details that make it a potent antibiotic.
And that's a theme we see again and again in polyketide biosynthesis. These post-assembly line modifications, they're essential for fine-tuning the activity, the stability, and the target specificity of many of these polyketides.
This is really eye-opening. Now, I'm curious, we've talked a lot about acetyl-CoA as the starting point for these pathways.
Right.
Are there other starter units that nature uses to create even more diversity?
That's a great question. Acetyl-CoA is the most common starter, but nature's got a few more tricks up its sleeve. Erythromycin, for example, uses propionyl-CoA as its starting point.
Okay.
And the oxytetracycline pathway, we just talked about that, it uses malonyl-CoA.
So different templates to kickstart the whole process.
Exactly. And the diversity of starter units doesn't stop there. We see isobutyryl-CoA in some pathways, and even more exotic examples like 3-hydroxypropionyl-CoA, which is used to make mupirocin, a topical antibiotic.
Wow. The variety is incredible. And I imagine these different starter units lead to some pretty unique polyketide structures.
Yeah.
What about those extender units? Are there any alternatives to the usual malonyl-CoA and methylmalonyl-CoA?
There are. Nature loves to mix things up. For instance, in the biosynthesis of swinnermycin, you have aminomalin and hydroxymalonyl SACP stepping in as extender units. These unusual building blocks, they contribute to the really unique structure and activity of this fascinating antibiotic.
It's like nature has an endless toolbox of molecular building blocks.
And we're only just beginning to understand how vast it is. With advances in bioinformatics and the fact that we can analyze so many microbial genomes now, we're uncovering more and more of these unusual starter and extender units, which is really exciting because it opens up all these possibilities for engineering new polyketides with very specific activities.
That is exciting. Now, before we move on to some specific case studies, I'd love to hear more about those post-assembly line modifications. You've mentioned them a few times, but I'm really intrigued by how they actually contribute to the final activity of these antibiotics.
Of course. Those post-assembly line modifications, they're absolutely essential for many polyketides to really reach their full potential. Think of them like the finishing touches that transform a rough draft into a polished masterpiece.
So what kind of modifications are we talking about here?
Well, they can be anything from simple additions like methylation, where a methyl group is just added on, or hydroxylation, where an oxygen atom is introduced, to more elaborate alterations like glycosylation, which is adding sugar molecules, and acylation, where osto groups are attached.
So it's not just about assembling the basic skeleton of the molecule. It's about decorating it, adding those crucial details that ultimately determine its function.
Precisely. Take tetracycline, for example. Those 9 enzymatic steps that transform that inactive pretetramid into the active form of oxytetracycline, they involve a very carefully choreographed sequence of methylations, hydroxylations, a transamination, and that final reduction step.
Okay.
These modifications, they fine-tune that molecule structure, making sure that it can actually bind to its target on the ribosome and have that antibiotic effect.
That level of precision is incredible. And I remember you mentioned the sugar additions play a crucial role in some antibiotics, right?
Absolutely. In macrolides like erythromycin, for instance, adding those specific sugar molecules, microrose and disosamine, that's actually essential for their activity. These sugar moieties, they act like guides, steering the antibiotic to its target on the ribosome and enhancing its binding affinity.
It's like a molecular GPS system.
Brilliant analogy. And in erythromycin, there's also a methylation step that modifies the microrose sugar. These modifications, combined with the ones that happen during the assembly process, transform that inactive DEB molecule into the potent antibiotic erythromycin A.
It's truly remarkable how nature has evolved these intricate systems to create these life-saving molecules.
It is a constant source of wonder. Now, to illustrate the sheer diversity of polyketide biosynthesis, let's delve into some specific case studies. In the next part, we'll explore the unique features of mupirocin and abyssamycin biosynthesis. These really highlight the incredible range of strategies that nature uses to assemble these fascinating compounds.
Sounds great. I'm ready to be amazed.
Welcome back. We're wrapping up our deep dive into the fascinating world of polyketide antibiotics. Ready for a couple more of those biosynthetic tales?
Oh, absolutely. I'm eager to hear what other tricks nature has up its sleeves. You mentioned some case studies, mupirocin and abyssamycin, right?
Right. So let's start with mupirocin. You might know it as Bactroban. It's that topical antibiotic that's often used for skin infections like impetigo. Now, here's what's interesting. Mupirocin, it's not assembled on one continuous assembly line, like erythromycin or tetracycline.
Okay.
It actually comes from two separate polyketide pathways working together.
So it's like two specialized factories collaborating to create that final product.
Exactly. One pathway makes this 9-hydroxin-ononoic acid moiety, which is kind of like a fatty acid. It uses this unusual starter unit called 3-hydroxypropenyl-CoA, and then it goes through 4 rounds of elongation, using malonyl-CoA as the extender unit.
Interesting. We've seen how those alternative starter units can really add a twist to these pathways.
For sure. Now, in this part of the molecule, all of the beta-ketone groups that are formed during each elongation cycle, they're fully reduced, just like in classic fatty acid synthesis.
Okay, so that part of the molecule is playing by the traditional rules. What about the other half of mupirocin?
That's where things get a little bit more elaborate. The other piece, called the monic acid moiety, is assembled on a type I PKS. It uses 7 modules spread across 2 proteins. And the initial product of this assembly line is a 14-carbon chain with 3 double bonds and 3 hydroxyl groups. But then, this linear intermediate, through a carefully choreographed series of enzymatic reactions, gets transformed into the rings that are characteristic of the monic acid moiety.
It's like those origami artists who can fold a simple piece of paper into a complex 3-dimensional structure.
Perfect analogy. So first, an epoxide, that 3-membered ring containing an oxygen atom, is formed, and then that epoxide undergoes a rearrangement to create a 6-membered pyrin ring.
Wow, those are some impressive molecular gymnastics.
It is, and there's another unusual feature in this pathway. During one of those chain elongation steps, a methyl group is added to a beta carbon, which isn't something we see all the time in polyketide biosynthesis.
So it's not just about adding carbons and oxygens, there's some strategic placement of other groups too.
Absolutely. These subtle additions can really impact the final molecule's shape and how it interacts with its target. Now, once both the monic acid and the 9-hydroxynonoic acid pieces are done, they're linked together through an ester bond, like 2 puzzle pieces fitting together. And this final coupling step is absolutely essential for mupirocin to actually work as a potent antibiotic.
So it's a true tag team effort, with each pathway contributing its own unique expertise to make this powerful antibiotic.
I like that. Now ready for another case study?
Yeah, bring it on. The more complex, the better.
All right, let's dive into the world of abyssamycin C. You might remember this one from our deep dive into folate biosynthesis.
Yes. I remember abyssamycin C being a really potent inhibitor of paraminobenzoate synthase, a key enzyme in that folate pathway.
You've got a great memory. And its biosynthesis is equally remarkable. Abyssamycin C, it has this very intricate tetracyclic structure. That means it has 4 interconnected rings. It's a true masterpiece of molecular architecture.
Okay, I'm officially intrigued. How does nature even begin to build such a complex molecule?
Well, like mupirocin, abyssamycin C is actually assembled in 2 parts that are later joined together.
Okay.
One part is an unsaturated linear polyketide, and the other is 5-membered tetronic acid ring. So the linear polyketide is made by a 7-module type I PKS, similar to what we've seen before. This PKS makes a 14-carbon chain with 3 terminal double bonds. That means they're at the end of the chain.
Got it. So we've got our linear polyketide. What about that tetronic acid ring? How does that come into play?
So that tetronic acid ring is synthesized separately and then attached to the linear polyketide. And the biosynthesis of this ring is quite unique. It involves 5 additional genes, which really highlights the complexity of this step.
Wow. A whole separate team of enzymes just to make that ring.
Exactly. And here's where it gets really interesting. It's thought that the enol part of that tetronic acid ring and those terminal double bonds of the linear polyketide, they undergo what's called a Diels-Alder reaction.
A Diels-Alder reaction. That takes me back to organic chemistry. That's that reaction where a dinon and a dienophile combine to form a 6-membered ring.
You're right on the mark. So in this case, the linear polyketide is the diorn, and the tetronic acid ring is the dienophile.
So they come together in this very specific way to create a new ring.
Precisely. And this deals all the reaction, it creates a tricyclic intermediate.
Okay.
So now we have 3 rings in the structure. But wait, the molecular acrobatics don't stop there. A tailoring oxygenase comes in. It specifically adds an oxygen atom to one of those remaining double bonds, forming an epoxide.
Ah, epoxides. They seem to pop up everywhere in these biosynthetic stories.
They do. And this epoxide is actually crucial in that final step of building abysomycin C. That epoxide undergoes an intermolecular ring closure, forming the 4th and final ring of the molecule.
So we've gone from a linear polytide and a separate tetronic acid ring to a very complex tetracyclic structure. Yeah. It's amazing how these pathways can do such intricate transformations.
It really is a testament to the power of those enzymatic assembly lines. Now, we've mostly focused on bacterial polyketide systems so far, but it's important to remember that fungi also make a bunch of different polyketides that have really remarkable biological activities.
Right, the fungal kingdom, a true treasure trove of bioactive compounds. So what makes their polyketide systems unique?
So fungal polyketide syntheses, or PKSs, are typically organized as single proteins that have all the catalytic and carrier protein domains. This is similar to type I PKSs in bacteria, but there's one key difference in how they operate.
Okay, I'm all ears. Tell me about that difference.
So unlike bacterial modular type IPKSs, where each module is used just once as the polyketide chain is being put together, fungal PKSs, they work in an iterative fashion.
Iterative? Does that mean they use the same module over and over again?
Exactly. It's like having this multi-talented worker on an assembly line who can do a bunch of different tasks.
That's a pretty efficient strategy.
Yeah.
Can you give me an example of an iterative fungal PKS in action?
Absolutely. A classic example is the biosynthesis of aflatoxin. That's a really notorious mycotoxin made by certain species of Aspergillus fungi.
Aflatoxin, the one that can contaminate food crops and be really dangerous.
That's the one. Aflatoxin biosynthesis involves an iterative PKS that makes this tricyclic compound called norsilurenic acid anthrone.
Cool.
And then this molecule goes through a bunch more modifications, ultimately forming those various types of aflatoxins.
So even though they operate iteratively, fungal PKSs can still make complex and biologically important polyketides.
Oh, absolutely. They might not have that clear-cut modularity of bacterial type PKSs, but they've got their own elegant ways to achieve structural diversity. And this brings us to one final fascinating twist in the world of polyketide biosynthesis hybrid systems.
Hybrid systems. Intriguing. What are those all about?
So these are systems that combine parts of both polyketide synthase, or PKS, and non-ribosomal peptide synthetase, or NRPS pathways. It's like merging 2 different assembly lines to make a whole new class of products.
Wow, that's incredible. So these hybrid systems can incorporate both amino acids and polyketide building blocks into a single molecule.
Exactly. And this fusion of pathways has given rise to some really amazing compounds with all kinds of structures and biological activities. A few well-known examples are bleomycin, an anti-tumor antibiotic, Yersinia bactin, a siderophore made by that plague bacterium, Yersinia pestis, and pristinomycin PII, a component of the antibiotic Synarcid.
Those are some serious players in the world of medicine and bacterial virulence. I'd love to hear more about how these hybrid systems actually work.
Okay. Let's focus on pristinomycin PII as an example. It's what we call a cyclic dipsipeptide, meaning it has both amide bonds, typical of peptides, and ester bonds, which are found in polyketides.
So a true hybrid molecule reflecting both pathways.
Exactly. The pristinomycin PII assembly line is a really cool mix of 6 PKS modules and 3 NRPS modules, all working together in a specific order. So the process starts with isobutyryl-CoA, one of those alternative starter units we talked about. Then 2 PKS modules extend the chain, followed by an NRPS module that incorporates the amino acid glycine.
So we've got polyketide and peptide extensions right from the start.
Exactly. Then the assembly line goes back to 4 PKS modules, adding more polyketide units, and then finally 2 NRPS modules incorporate the amino acids serine and proline.
Wow. What a complex choreography. So how does that final cyclic structure actually form?
The chain is released, and it cyclizes by forming a lactone bond between that starter unit and the proline residue at the end of the chain. This lactonization step is really important for pristinomycin PII's activity.
So it's similar to the macrocyclization that we saw in erythromycin, But in this case, it's using an ester bond rather than an mi bond.
Exactly. And those two stretches of polyketide units within pristinomycin PII, they actually contribute 3 double bonds, a hydroxyl group and a ketone group, to the final molecule. And then that serine residue, it undergoes a cyclization and an aromatization reaction, forming this heterocyclic oxazole ring. Plus, that proline residue, being a cyclic amino acid, adds even more rigidity to the structure.
It's a really remarkable blend of linear and cyclic elements, all coming together to create this very specific and constrained three-dimensional shape.
Precisely. And this constrained shape is absolutely essential for pristinomycin PII to bind to its target on the ribosome and do its job as an antibiotic. Now, one of the challenges when studying these hybrid systems is figuring out how those PKS and NRPS modules communicate and coordinate their activities. They need to hand off that growing molecule smoothly, recognize each other's substrates, and make sure everything happens in the right order.
That makes sense. It's like having workers from 2 different factories collaborating on a project. They need to be able to speak the same language.
Right. And researchers are still figuring out all the molecular details of how that coordination actually works. But we know that those key catalytic domains, the ketosynthesis or KS and PKS modules, and the condensation or C domain and NRPS modules, they have to be a little bit promiscuous.
Promiscuous. Okay, that's an interesting way to put it. What does it mean in this context?
It means that those enzymes need to be a bit more flexible in their substrate preferences compared to their counterparts in purely PKS or NRPS pathways. So, for example, a C domain in a hybrid system needs to handle both amino acid and polyketide substrates, even though it normally only works with 2 amino acids. Similarly, that KS domain needs to be able to accept a peptide substrate, even though it's used to working with polyketide chains.
So they have to be adaptable, able to recognize and work with a wider range of building blocks.
Exactly. And that adaptability is what makes these hybrid systems so incredibly powerful. It opens up a whole new world of possibilities for creating molecules with unique structures and properties that wouldn't happen with just one kind of assembly line.
This has been an amazing journey. I had no idea there was so much complexity and ingenuity going on inside these little microscopic factories.
It is truly fascinating, and we've only just scratched the surface. With the world of microbial genomics constantly expanding and all those bioinformatics tools we have, who knows what other incredible pathways and molecules are waiting to be discovered.
Okay, before we wrap up, I have to ask. Remember at the beginning we said this deep dive is based on Chapter 16 of Antibiotics, Challenges, Mechanisms, Opportunities, by Walsh and Wensibs, and there was something in there about exam questions.
Yes. Let's put your polyketide knowledge to the test. But don't worry, these are just for you, our listener, to see how much you've learned.
Okay, I'm ready for the challenge.
All right, first question. What key chemical difference distinguishes chain elongation in polyketide synthases, or PKSs, from non-ribosomal peptide synthetases, or NRPSs?
Let me think back. I remember it has something to do with the type of bond that's formed during elongation. PKSs create carbon-carbon bonds, while NRPSs form nitrogen-carbon bonds. Am I right?
You nailed it. That's the fundamental difference between those two awesome assembly lines.
Okay, feeling good so far. What's next?
All right, question 2. How does the presence or absence of tailoring enzymes, those KR, DH, and ER enzymes within PKS modules, contribute to the structural diversity of polyketides?
Okay, this takes me back to our discussion about incomplete reduction. Those tailoring enzymes are like gatekeepers. They determine the level of reduction that happens at each step. So if a KR is missing, the ketone group sticks around. If the DH is absent, the hydroxyl group remains. And if the ER is inactive, you end up with a double bond in the polyketide chain. So basically, nature can create a huge variety of polyketide structures with different functional groups and properties just by mixing and matching those enzymes.
Exactly. You've got it. You really understand how those tailoring enzymes fine-tune that final product. Ready for question 3?
Hit me with it.
Okay. Describe the mechanism of chain release and macrolactone formation in type I modular PKSs, like the ones that make erythromycin.
All right, focusing on the grand finale. I remember it involves a special enzyme, a macrocyclizing thioesterase, or TE. It's a 2-step process. First, the TE moves that growing polyketide chain from the ACP to its own active site. Then it keeps water out and positions a specific hydroxyl group on the chain so it can attack the thioester bond, and that forms the ring and releases the mature macrolide.
Perfect. You've described that mechanism beautifully. Question 4. What makes type 2 PKS systems different, like the ones used to make tetracycline?
Okay, let's see. Type 2 PKS systems are those simpler ones, organized like independent workshops. The main difference is that they often don't have those tailoring enzymes, the KR, DH, and ER, that are so important in the modular type I systems. And this lack of tailoring enzymes is what leads to those reactive polyketone intermediates that like to cyclize. And that's exactly how those characteristic rings in tetracycline are formed.
Spot on. You've highlighted all the key features of type 2 PKSs. Ready for the final challenge.
Bring it on.
All right, last question. Give me an example of a hybrid NRPS PKS assembly line product and explain how these hybrid systems contribute to all that diversity in natural product structures.
Okay, let's go with pristinomycin PII, that cyclic depsipeptide that has both amino acid and polyketide building blocks. These hybrid systems are like the best of both worlds, letting nature combine building blocks and tailoring reactions from both PKS and NRPS pathways. And this leads to molecules with totally unique structures and properties that wouldn't be possible with just one type of assembly line. It's like having a team of chefs from different culinary traditions coming together to make a totally new dish.
Fantastic. You aced all 5 questions. You're Polyketide Pro.
I feel like I've earned an honorary degree in polyketide biosynthesis.
And to celebrate, I have a little something to share. Remember how we were talking about polyketides being like intricate works of art? Well, I tried to capture that feeling in a short poem.
Oh, I love it. Please share.
Okay, here goes. In microbial realms where wonders reside, molecular machines with nature as guide, polyketides arise, a symphony of form from simple units, a potent storm. Acetates dance, a chain begins to grow, beta ketones join, a vibrant show. Tailoring enzymes with artful precision sculpt the structure, a wondrous vision. Modular or iterative, the pathways entwine, type I and type 2, a grand design. Hybrid systems merge, a fusion so bold, peptides and polyketides, stories yet untold. Macrolactone rings, majestic and grand, constrain conformations as nature has planned. Sugars adorn with elegant grace, guiding the molecules to their rightful place. From muparosin's touch to abyssamycin's might, polyketide antibiotics, beacons of light, microbial factories, a treasure to behold, a testament to nature's stories yet to unfold.
That was beautiful. It really captures the elegance and power of these biosynthetic pathways.
Thank you. It's been a pleasure diving deep into this world with you.
And with you, our listeners. We hope you've enjoyed exploring the amazing world of polyketide antibiotics. It's a true testament to the ingenuity of nature, a source of inspiration for us scientists, and ultimately a beacon of hope in our fight against infectious diseases. So until next time, keep those minds curious and those questions coming.